Assalamualaikum and good morning po, ma'am. So my report today is the human genome. So I have my outline in my report, which is what is a genome, what made up of genome, what is genome sequence and how it work, what is human genome project. So first, let us know what is a genome. So genome is all the genes that make up an organism. Genes are made up of DNA. DNA is made up of paired strands. So all organisms have their own genome. Each genome contains that information needed to build and maintain that organism throughout its life. So our genome is the operating manual containing all instructions that help you develop from single cell into person you are today. It guides your growth, helps your organs to do their jobs, and repair itself when it's damaged. So, genes is a segment of DNA that provides the cell instruction for making specific protein, which then carries out a particular function in our body. Genomes are complicated, and while a small number of traits are mainly controlled by one gene, most traits are influenced by multiple genes. So, DNA is read like a code. The order of letters in this code allows DNA to function in different ways. The code changes slightly from person to person, helping make who you are. So that code is the adenine, thymine, guanine, and cytosines. So genomes are made up of DNA, an extremely large molecule that look like a long twisted ladder. This is iconic DNA double helix that you may have seen in textbooks or advertising, or we have a picture in there. So the DNA in a cell is not a single long molecule. It is divided into a number of segments of uneven lengths. So every fungus, plants, animals, and had have a set number of chromosomes. For example, humans have 46 chromosomes, 23 pairs, and rice plants have 24 chromosomes, and dogs have 78 chromosomes. So, meaning to say, a genome is organism complete set of DNA including all of its genes. Each genome contains all of the information needed to build and maintain that organisms. So, we have picture in there. So, the next picture is our genome. So, we have here examples that father, mother. The role of our parents is necessary for you to grow throughout your lifetime are passed down from your mother and father. Half your genome comes from your biological father and half of it is your mother. So making you related to each other but identical neither. Your biological parents' genes influence traits like height, eye color, and disease risk that make you unique persons. So we have two types of genomes. They are the prokaryotic genomes and eukaryotic genomes. So, the prokaryotic genome are mostly single circular chromosomes, while eukaryotic genomes consist of one or two sets of linear chromosomes confined to the nucleus. So, what is a genomics? Genomics is mapping and sequencing to analyze the structures and organ. Genomics is the study of structures and function of whole genomes. So, meaning genomes is focusing on structure, function, evolution, mapping, and editing our genomes. So, the genome sequence. Genome sequencing is the technique that allows to read genetic information found in anything from bacteria to plants to animals. Sequencing involves the determining the order of bases, the nucleotide subunits adenine, guanine, cytosine, and thymine. So the sequence information of genome will show the position of every gene along chromosomes. We have picture in there, and the regulatory regions that flank its gene, and the coding sequence that determines the protein produced by each cells. So sequencing DNA means determining the order of four chemical building blocks called bases, which is the adenine, guanine, cytosine, and thymine that make up DNA molecule. That sequence tells scientists that kind of genetic information that is carried in particular DNA segment. So, steps of genome sequence. Genome into smaller fragments, sequence the smaller pieces, sequence of short fragments together, DNA sequencing approaching hierarchical, shotgun sequencing is useful for sequencing genome of higher vertebrates that contain repetitive sequences. So we have picture in there, letter A and B. So A is the whole genome shotgun sequencing, useful for smaller genome. So letter B is hierarchical shotgun sequencing, useful for sequencing genome of higher vertebrates that contain repetitive sequence. So during whole genome sequences, researchers collect a DNA sample and then determine the identity of the 3 billion nucleotides that compose the human genome. The very first human genome was completed in 2003 of the part of Human Genome Project, which was formally started in 1990. So DNA, we have DNA and proteins. Genome contains just over proteins coding genes. Far fewer had protected. In fact, only about 1.5% of genome codes for protein, while the rest consists of non-coding RNA genes, regulating sequence, and introns. However, a single gene can produce a variety of proteins by means of RNA splicing. One particular Dropelia genes can be alternative spliced into 38,000 different mRNA 9. So we have Human Genome Project. So the Human Project was one of the great feats of exploration in history. Rather than outward exploration of planet or the cosmos, the HCP was inward voyage of discovery led by international team of researchers looking to sequence and map all of the genes together known as the genome of members of our species Homo sapiens beginning on October 1, 1990. And completed in April 2003. So in humans, a copy of entire genome, more than 3 billion DNA base pairs, is contained in all cells that have nucleus. The Human Genome Project was the international research effort to determine the DNA sequence of entire human genome. So we have difference between human chimpanzees. Kumbaga, kasi sa atin kasi parang mas malapit daw tayo sa chimpanzees kasi more sa genome ng chimpanzees is parang nafa-found out din sa ating mga katawan. Tulad niyan, difference between human and chimpanzees. The animal that is alive now is closest to human is the chimpanzees. 98% of the DNA is the same between humans and chimpanzees. So, thank you po, ma'am.